DNA in the Cell Cycle, G0, G1, S, and G2, by Laureano Andrade Vicenti, MD, and pre-med disciples.
Here we are on this topic list here. You'll note that we're not going to go through mitosis, and you'll note that in the title I didn't say mitosis. And that's because I'm not going to go through mitosis in this lecture. I just have too much to cover, and it'll just be too long of a lecture. But the rest of the stuff we're going to go through. We'll have a lot of fun doing it, so let's get on to it. We'll start off with structure of the nucleic acid, then we'll go through function, then we'll go through DNA replication, and some repair mechanisms, and some things in between. So why don't we get on to it now. DNA is found within the nucleus. Okay, within the nucleus is where DNA is found. Okay, however, you can also find DNA within the mitochondria. They have circular DNA, and that denotes they are passed in the so-called and provide support for the endosymbiotic theory that says that we engulfed an aerobic microbe and we did a horizontal gene transfer. That is, we transferred the genes that are within the mitochondria and into then our nucleus, and over time that became an organelle as opposed to an endosymbiote. So he lost his autonomy, if you would, and became dependent on us so much so that we just called it an organelle instead of another organism. Anyway, DNA is found within the nucleus in the mitochondria. The central dogma of biology said that we go from DNA to RNA to protein. Okay, DNA to RNA being transcription, RNA to protein being translation. Over the course of a few lectures, I'll be discussing each one of these processes, but today we'll be dealing with exclusively DNA. And in fact, we'll be dealing with DNA in the context of DNA replication, which then clearly is just the replication of our existing DNA. So, without further ado. So here is a DNA, double-stranded DNA. This is DNA interacting with one another. What you can find here, what is easily notable, the phosphate backbone that makes up DNA, the ribose sugar. So what makes this a sugar is the fact that it's a heterocycle containing oxygen and has a bunch of OHs coming off of it. And you have the glycosidic bond connecting the ribose sugar to the nucleobase, which is labeled here. Okay, and they are hydrogen bonding with another strand of DNA running in the opposite orientation. You can also note that I've labeled this as 5' to 3' and 3' to 5'. That is the way in which DNA anneals to another strand of DNA. They run in opposite orientations. You may not be able to yet appreciate what those numbers mean, but you'll gain an appreciation shortly. So what you do, if you take this DNA strand and you twist it into a double helix, so essentially twist it around another twist, that's why it's called double helix. These DNA strands, this is the phosphate backbone, then like orange and red here, and then we have the nucleobases here interacting with one another, hydrogen bonding and base stacking with one another. Okay, and notice it has a major groove and a minor groove. This is major groove, this is minor groove. And you'll get a taste about what we're going to go through each little piece of this DNA. So we'll leave it at that for now, and we'll go through each little piece. We should talk about what makes up DNA. What makes up DNA is actually nucleotide monophosphates bound up together. Okay, so let me describe for you essentially then the building block of DNA. I'll have to describe each part here. What we have here, the nucleobase. Okay, this portion right here, the nucleobase, or the base is another word for it. Then you have the nucleoside. So the nucleoside is actually consists of the nucleobase bounded to then a ribose sugar by a so-called glycosidic bond. A glycosidic bond is just a bond between a sugar and another molecule. And no phosphate group. So that's what a nucleoside is. However, a nucleoside is not the building block of DNA. A nucleotide is the building block of DNA. So once you add a phosphate to a nucleoside, it's now called then a nucleotide. Right, and you can have nucleotide monophosphate. In this case, it's adenosine because this is actually an adenosine base. All right, or you can have adenosine triphosphate, adenosine diphosphate, and adenosine triphosphate corresponding then to the more phosphate groups there. This is what the adenosine monophosphate is what makes up then DNA. Well, nucleotide monophosphate could be A, T, G, or C. So what we'll do first is we'll describe the nucleobases, and then we'll describe the nucleosides, the nucleotides. Let's start with nucleobase. So the nucleobases that make up either DNA or RNA are the pyrimidines and the purines. The pyrimidines are one-ringed heterocycles, aromatic heterocycles, and the purines are two-ringed aromatic heterocycles. What make up the pyrimidines are cytosine, uracil, and thymine. I like to put them in that order to remember cut. Just for cut the pie, the mnemonic to remember that those are the pyrimidines. You can also remember that pi is one circle, so cutting the pie, all the pyrimidines are going to have one circle. Again, these are just aromatic heterocycles. The purines, again, are two-ring aromatic heterocycles containing nitrogen. And the mnemonic is pure as gold to remember that the purines are adenine, and guanine. Okay? And you bring two rings to the wedding, bring two rings to the wedding, and so that'll help you recall that these are two-ring structures. Watson and Crick are the guys who, quote-unquote, discovered the structure of DNA, quote-unquote now. And they said that the base pairing is as follows. A paired with T or U, and G paired with C. You'll get a picture of that shortly. However, we'll first look at the nucleoside as well as the nucleotides next, and then we'll move on to then the base pairs and such. So, here is an example of a nucleotide. Okay, again, I told you nucleoside is just minus the phosphate groups. That's what you'd have a nucleotide for. This is an example of deoxyriboadenosine triphosphate, or DATP. Okay, if it was not adenosine or if you didn't want to be specific about what nucleobase was there, you would call it DNTP. Okay, the D is corresponding to then the fact that there is no hydroxyl group deoxy at this 2' position. And I haven't discussed this nomenclature with you yet. There's 1', 2', 3', 4'. So in organic chemistry, the way in which we number carbons is we just number them 1, 2, 3, 4, 5, 6, 7, etc. However, our nucleobases already have that numbering system. So in order for us to differentiate between the nucleobase and the ribose sugar in terms of carbons, we had to number the ribose sugar as 1', 2', 3', 4', 5' instead of 1, 2, 3, 4, 5. And that's just for us to be able to differentiate and talk about it easily. So that's the reason why we call it 1', 2', 3'. This here is the 3' position. You'll be hearing a lot about this throughout the lecture. The 3' position is very important because it is what allows us to extend DNA. That 3' OH at the 3' position is what allows us to extend and attack another incoming NTP to then form a new phosphodiester bond. Anyway, we'll get there. That's DATP. That is what's found in DNA. Okay, deoxy. Now, that's what's found in DNA. How about RNA? So this is what is the nucleotide that we use to then ultimately form RNA. And that is going to be riboadenosine triphosphate. That's the name. Or you could call it ribonucleotide triphosphate if you didn't want to specify adenosine as the base. And that's called R-ATP. Notice the difference between DATP and R-ATP is just that we don't have the deoxy prefix because we actually do have a hydroxyl group at the 2' position. Whereas in the DNA, we don't have it, so we call it deoxyribo. But that's the difference. So, and that is what makes RNA RNA. It had nothing to do with the uracil base. You can have a uracil base in DNA. You don't normally have it, and it's recognized as abnormal, but that's not what makes it RNA. What makes it RNA is the 2' OH. You also may see the terminology DNTP in the context of research, okay, and research-based methods. So that just means deoxyribonucleotide triphosphates, and RNTP just means ribonucleotide triphosphates. Oftentimes you'll just see ATP or GTP or CTP or UTP, and that's just the researcher not specifying which, what kind it is, because it's fairly obvious which kind it is. It's fairly obvious that if you're doing DNA replication, you're going to use DATP. So sometimes they'll just write ATP, or sometimes they'll just write NTP, because it's fairly obvious is what you're doing. All right, we should probably go over a little bit more nomenclature. So next slide here. The nomenclature associated with nucleotides and nucleosides, and you should know it. So for a nucleoside, let's say you have the base uracil and you have a nucleoside, so you don't have any of these phosphate groups. You would call that, instead of calling it uracil, in order to denote that it's a nucleoside and not just a nucleobase, you would call it uridine. If it was a nucleotide, so you added the phosphate groups, then you would call it uridine mono, di, or triphosphate. So this is an example of uridine triphosphate. Right? You could also do cytidine triphosphate. You could do thymidine triphosphate. Again, the original nucleobase name was cytosine, and the original nucleobase name was thymine, but you convert it as follows, that nomenclature. The purines do a similar thing. All right, for nucleoside, you change the ending to sine. So if you have adenine, if it's a nucleoside, you call it adenosine. And if it was guanine, then in a nucleoside form or nucleotide form, you call it guanosine monophosphate, let's say. And that's pretty straightforward. All right, so some more nomenclature that you should know, just in case you run into it within a passage. A nucleic acid is just a polymer of nucleotide, i.e. DNA or RNA. Double-stranded DNA, dsDNA, double-stranded DNA. Single-stranded DNA, self-explanatory. mtDNA is mitochondrial DNA. cDNA is complementary or clonal DNA that has been reverse transcribed from RNA. We haven't talked about that, but reverse transcription is just RNA to DNA. So going back towards DNA from RNA. That's reverse transcription. And if you get DNA from that, you call that DNA cDNA. If you see any other DNA names, you can often deduce what they mean just from the passage, or from prior knowledge, or just from common sense. Like, this is clearly single-stranded DNA. This is clearly double-stranded DNA. So, just use your head if you see anything else. Alright, so why don't we take a closer look at the base pairing that's going on within DNA. If you zoom in on this a little bit, and I know we didn't zoom in on it too much, what you'd find is hydrogen bond donor, then hydrogen bond acceptors interacting with one another. This is the hydrogen bond donor, show it clearly because it has hydrogen connected to FO or N, and this is the hydrogen bond acceptor. And you'll find that these interactions then will allow for then, in part, the formation of DNA, because it is these interactions that stabilize the formation of DNA in part. Again, there's another piece to it. And if you don't have these interactions, so you have a mismatch. A mismatch is just the fact that you have electron acceptors or you have another electron acceptor, then they have electronic repulsion and they mismatch. The distances become longer than what they should be. So you'll find that if you look at the bonding distance from N to N on each nuclear base, you'll find that it's about 11 angstroms average, whether you're talking GC or AT. That is important, okay? First off, it's just structurally important for the DNA to be around that size. It's just the optimal bonding distance. And the other part of that, you can't really catalyze the replication of DNA if these bonding distances are out of register. Because as you'll come to appreciate, this 3' OH is what's important in catalyzing the formation of new DNA and the polymerization of nucleotide into nucleic acid. One last comment that I just want to make is that if you ever ask the question, well, what takes more energy, to break up a GC bond or an AT bond? Use your head. The GC bond has more hydrogen bonds. It has more favorable interactions between the G and the C than does an A and a T. So a GC bond is going to take more energy to break a bond than, let's say, an AT bond. Okay. Another comment that I want to make is that I mentioned that there's more to it. The structure of DNA, there's more to the structure of DNA than just the hydrogen bonding between the nucleobases. The nucleobases undergo also a thing called base stacking. So base stacking is just essentially the hydrophobic effect in action. Base stacking, or the bases actually being on top of one another, one against the other, and interacting positively, is actually more of not so much an energy effect, not so much a van der Waals positive interactions effect, but rather an entropy effect. The base stacking will actually maximize the entropy of the surrounding water molecules that have to surround the DNA. Whereas if, let's say you had your base out here, and I'm just going to redraw a base like this. Let's say you had your base out here, and some of the bases in here, then you wouldn't get the base stacking. And actually, water molecules, and I'll just represent them as little circles, would have to surround this base, alright, the hydrophobic base, and actually have to form ordered water cages around this water molecule. And that'll actually decrease the entropy of water. And so to maximize the entropy of water, the bases actually stack up on each other like this inside the DNA helix. You probably don't get that unless you've watched a thermodynamics lecture, so it's okay if you don't get it yet. So that brings us then into a conversation about Chargas rules. It's nothing more than logic, really. It states that in double-stranded nucleic acids, DNA, RNA, the percent A is equal to the percent T, or the percent A is equal to percent U if you're talking about RNA. And it also says the percent G is equal to percent C. Again, that's completely logical. If at every time you have an A on one side, you have to have a T on the other, then clearly the percentage of A you have in that solution is going to be equal to the percent T you have in that solution of DNA. Of double-stranded DNA, rather, not the solution. The solution may have other A's and T's in solution, but of the DNA itself, of the double-stranded DNA itself, or RNA. So then, Nothing really simple there except to do this exercise. So I'll read it to you. Exercise 1, you collect a sample of an unknown microorganism that you suspect to be a double-stranded RNA virus. You sequence the genome of the unknown microorganism and determine its genome has the following ratios. It has 30% A, 20% G, U 30%, T 0%, C 20%. Is this microorganism likely a double-stranded RNA virus? Yes, no, or there's no way to tell. Feel free to pause and think for a second. The answer is yes, right? It had uracil, which doesn't make it RNA per se, but uracil is characteristic of RNA. And it had the right percent composition, so 30, 30, 20, 20, to follow Chargas rules to make it double-stranded nucleic acid. So yes, it's likely a double-stranded RNA virus. Exercise 2, given the following data, determine the percent composition of each nucleotide in a double-stranded DNA virus. So feel free to do that. I'm not even going to fill that out for you. Actually, I lied, because if I don't fill it out for you, then you're just going to ask me about it. 15. Okay. I leave this with 70% left, so we have 35 here, and we have 35 here. There you go. And of course 0 for the uracil because this is double-stranded DNA virus. There you go, there you go. Alright. Again, I mentioned this earlier. I said, why doesn't A pair with C? Why doesn't G pair with T? Well, the reason is because you got electronic repulsion when those happen, when they try to pair with one another. Electronic repulsion. Electronic repulsion. Remember, Coulomb's law is K big Q little q over R squared. If you're the same charge, you're going to get repulsion. If you're opposite charge, you're going to get attraction. They're going to repulse each other. First off, these bond distances are going to be huge. And then you'll get a bulge with some match, and you'll see it'll appear like this. This is one way that we appears. And that's just due to the mismatch. Due to that, the bond distances become longer than normal. It'd be like this. Because of the fact that you had an AC instead of an AT. As you know, that has problems for enzyme active sites. So, in the beginning of the lecture, I told you that this is what DNA looks like. But the reality of it is, it's kind of more complicated than this. In the sense that when you look under the microscope, you don't see this. You actually see something more than this. They're actually proteins that are bound up with DNA. Those proteins are called histones, and that's what we'll go through next. DNA will actually... So you take DNA, deoxyribonucleic acid, this polymer, and you wrap it around protein. Once you wrap it around protein, you can call it chromatin. Okay, so any of these things, structures here that I'm going to show you, that's all just called chromatin. Now, the DNA wrapped around the histone protein, those are just the proteins, it's an octamer of protein, that's called the nucleosome. So this little structure here, this circle with DNA around it, that is called the nucleosome. And those nucleosomes can pack up together to form these so-called beads on a string structure. And those beads on a string structure are genes that are going under expression, so transcription. We haven't gotten there yet. And then you can link those up, okay, you can link those up by histone linker proteins to then form this tightly packed structure. Now this tightly packed structure undergoes less transcription. So this is what would be called then heterochromatin. And this is more likely called euchromatin. So heterochromatin is really tightly packed DNA, chromatin. And euchromatin is loosely packed chromatin. Euchromatin is associated with high expression of genes. Heterochromatin is associated with low expression of genes. Again, chromatin is just DNA plus the DNA binding proteins, the histones. So let me show you the structure of a nucleosome. So a nucleosome consists of an octamer of core histones. You know, they have names, H2A, H2B, H3, H4. Let's not go there. They just have an octamer of core histones. And DNA wraps around those histone octamers to fit into the nucleus. DNA by itself is really, really long. Really, really long. The genome contains about 3 billion base pairs of DNA. Okay, the nuclear genome contains about 3 billion base pairs. Those base pairs, you have to fit that in a very small, on the order of micrometers, the distance the nucleus is, about 6 micrometers in diameter. And so there's not a lot of space to work with. And so the DNA really has to fold up very tightly to fit into the DNA. And that's what they do with these histone binding proteins and the H1 linker histone linking up these little beads on a string to form this less active fiber, this 30 nanometer fiber structure. These core proteins contain tails, N-terminal tails, which can be modified. And that when you start to modify histone proteins, when you start to modify DNA, and you don't change the base pair, that's called epigenetics. We're going to get there. Okay. You can either modify them via acetylation or methylation, and there's some other modification, but let's deal with acetylation and methylation. So acetylation, what that'll do, it'll tend to decrease packing and hence increase gene expression. And if you're wondering why, well, think about the structure of DNA. All right, you get that phosphate backbone. That phosphate backbone is negatively charged. Okay, so if you acetylate something, you're gonna have a negative charge. Okay, that acetyl group is just a CH3 CH double bond oxygen group. Okay, so something like this. I'll show you in the next slide. That acetyl group, the oxygen from the acetyl group will actually repulse the phosphate backbone, and you get decreased packing. Because again, that's just Coulomb's law. If you methylate, you're going to increase packing and decrease gene expression just from kind of hydrophobic effect. They're going to pack together. The hydrophobic molecules are going to pack together more tightly. Or the other way to think about it, now you have an uncharged molecule as opposed to a partial negative charge. And why does packing affect transcription rate? Again, I told you earlier. If you pack more tightly, then the transcription factor and the RNA polymerase to do transcription, the machinery, cannot reach the area or have a tougher time reaching the area, so you get less gene expression. Whereas if you pack less tightly, then the transcription factor, then the RNA polymerase and all that jazz can get to the promoter region and transcribe the locus. So let's just go through that. So if you want to acetylate the tail, so this is the tail. This is that tail I told you. It's just a string of amino acids. These are the one-letter codes for the amino acids. This happens to be a lysine residue. And HATs, histone acetyltransferases, are proteins that will catalyze the transferring of an acetyl group to the lysine residue. A big target is often H3K9. So the acetylation of H3K9 will cause for a looser packing and increased gene expression. All right. Histone deacetylase, the HDACs, will just remove that acetyl group. So I just realized I didn't explain what H3K9 means. So H stands for histone, the 3 stands for H3, that H3 histone, so this one. And the K means the 9th lysine residue. So if you count from here, from the N terminal, you'll find that you go 1, 2, 3, 4, 5, 6, 7, 8, 9. The 9th residue, so K9, lysine. The K stands for lysine. That's this one-letter code. You can also methylate the position. That'll be catalyzed by histone methyltransferases. All right, it'll methylate that group, and it'll cause tighter packing. Okay, it'll cause tighter packing. You can also methylate the actual DNA itself. Again, that gets into the realm of epigenetics. What will happen is there'll be a protein, a histone protein 1, that'll bind to methylated lysine residues. Okay, and that'll be then serve as the scaffold for a DNA methyltransferase, and then that'll catalyze the methylation of DNA, particularly usually cytosine. All right, and then what'll happen then, and then that can actually spread. That silencing effect can actually spread to neighboring DNA, because now a scaffold protein, a DNA binding protein, can then bind that methylated cytosine residue, and then a DNA methyltransferase can bind and spread the methyl group to the adjacent portion of DNA. That's the interesting part of it. Either way, you get tighter packing. Again, this is just making the point here that euchromatin is less tightly packed and heterochromatin is more tightly packed. Again, heterochromatin under electron microscopy will look dark under the microscope, whereas euchromatin will look lighter. And again, it's less tightly packed, so the transcription machinery can get there and bind, whereas in heterochromatin, you get a decreased gene expression because it's much harder to reach that area. Turns out that heterochromatin is typically found toward the periphery of the nucleus, whereas euchromatin is found more towards the center. I'm just going to do a review about epigenetics. Epigenetics are changes to the genome that do not change the base pair. That's essentially what we just went through. Okay, for example, acetylation and methylation of the DNA, and to provide the way in which the environment can be inherited. However, now, most of the time when you get methylation of the DNA in the genome, it's not passed down from parent to offspring. However, some genes are, and those are the genes that we're going to go through next. That's paternally and maternally imprinted gene. So a paternally imprinted gene is turned off in sperm, and a maternally imprinted gene is turned off in oocytes. I'm going to leave it at that today, and you're going to get this a few times in a few different lectures, so I'm just going to leave it at that. I don't want to confuse you. Alright, so we left here in terms of packing, we left it at this area here. So this is under normal cellular conditions what it looks like. If you're getting a lot of transcription, it looks like this. If you're not, you're going to be in your heterochromatin form, and you're going to look like this, like these packed up 30 nanometer fibers. But when you go through mitosis, and when you go through, you start going through the cell cycle, particularly mitosis, you're going to pack up into what are called chromosomes. Okay, so the chromatin is going to type very, very tightly. It's going to add more scaffolding proteins and eventually form a chromosome. A chromosome is an extremely tightly, densely packed structure of chromatin. All right, that's all it is. And here's what they look like. Here is a chromatid. So a chromatid is just unreplicated DNA. And if you put two chromatids together, you're going to get a chromosome. Where you get the second chromatid is via DNA replication. You replicate DNA to get that second chromatid, and then those are called sister chromatids. They're called sister chromatids because they are identical to one another, and they came from each other, and they are like twin sisters. So they're called sister chromatids. There's a nomenclature associated with the chromosome. So the P arm is the shorter arm, the Q arm is the long arm, and the tips are called telomeres. We're going to go through that at the end of the lecture. And you also have the centromere. It's a long tandem repeat structure, and it's the site of constriction. Again, we're going to go through what a long tandem repeat is, but essentially what it is just a repeating sequence of DNA. We're going to go through that terminology and everything, so don't worry. Here is a karyotype. So a karyotype is just a right chiasma stain of the chromosomes when they're about to go through mitosis. These are chromosomes that have already been replicated. So actually, each chromosome is one of these little strings, but that actually makes up two chromatids. And each little pair is a pair of chromosomes, and actually makes up 4 chromatids. The autosomes are 1 through 22, and they're found in both males and females at the same rate. And then you have the sex chromosomes, which are X and Y. XY for male, XX for female in humans. The little bars that you see, just the right chiasma stain being taken up at a differential rate depending if you have GC-rich areas or AT-rich areas. And the chiasma stain is just a stain. It just stains things. It gets taken up by the molecule. Let's not go into that histology. Anyway, you find chromosomes when you're in cellular division. Under normal conditions, you won't see this on a microscope. If the cell is not undergoing mitosis, you're not going to see this. You're just going to see chromatin. We have 23 pairs of chromosomes, or 46 chromosomes altogether. Half from mom, half from dad. Karyotypes show, again, this is what I said, karyotypes already show replicated DNA. And that's part of why people get confused about this. What's a chromosome, what's a chromatid? Well, I told you. So, one of the other different types of karyotypes you might see is where they actually use fluorescent in situ hybridization. So that's how this is worked. You take a probe DNA. A probe DNA is typically just an oligonucleotide DNA that will then bind to your target DNA. What you do is you take your probe DNA and you label it with a fluorescent dye, so or a fluorophore. Okay, that the fluorescent dye, the fluorophore, will fluoresce it will fluoresce at any time. So what you need to do is you need to make it bind to your target DNA by just putting it in solution. And then it'll fluoresce wherever your target DNA is. And if you're doing it in a chromosome, so a very condensed chromatin form, you'll see it under the microscope in the chromosome form and fluorescing. Nothing special there. I also want to go through this terminology: homologous versus non-homologous chromosomes. Homologous just means that you're similar in position, structure, and evolutionary origin, but that doesn't really help you much for this. Alright, so let me go through this. Say we have chromosome A from dad and chromosome A from mom. Since they're both chromosome A, okay, they're both going to have, let's say, gene A right here and gene A right here. However, they're not identical. They're in the same position. All their locuses are in the same positions. All the genes are in the same positions. But they may have a few different nucleotides in a few different places. So if this was gene A, this may be gene A allele A, and she might have gene A allele B here. Okay? So they're not completely similar. They're very similar, but they're not identical. Where the sister chromatids are identical. This is an exact copy of this because this arose from DNA replication. Okay? However, these are just homologous to one another. These may have different alleles. Okay? Although the same genes in the same places. Okay? So those are homologous chromosomes. Chromosome A from dad is homologous to chromosome A from mom. I think you can see that they're similar in structure, position, and evolutionary origin because of that. Whereas you can have non-homologous chromosomes. So a non-homologous chromosome would be, let's say, chromosome A from mom and chromosome B from dad, or chromosome A from mom and chromosome B from mom. Okay, those are non-homologous because they're not similar in position, structure, or evolutionary origin. Okay, they don't have, this has gene A here, but this doesn't have gene A anywhere. This has just a bunch of different genes. This has a bunch of different genes compared to chromosome A from mom. So they're non-homologous. Okay, they don't have gene A anywhere, and this doesn't have gene B anywhere. And I'm just using this terminology superficially. But these, however, are sister chromatids to one another, and these are homologous chromosomes to one another. Chromosome B from dad rather, is homologous to chromosome B from mom. Because again, they have gene B, let's say right here, and she has gene B right here. And although they may be a slightly different sequence, maybe have 1 or 2 different nucleotides, or 3 different nucleotides, 4 different nucleotides, it's the same gene in the same place, similar position, structure, what have you. And that's the difference between homologous and non-homologous chromosomes. Homologous chromosomes are just the same chromosome from different parent, whereas non-homologous chromosomes are different chromosomes and may be from different parent or from the same parent, just different chromosomes. So if we're thinking autosomes, the chromosomes from autosome 1, the chromatids are sister chromatids, and then the pair are homologous chromosomes, but the autosome 1 is a non-homologous chromosome to autosome 2. There's some other representations of DNA. You may see A, P, T, P, G, C, P, G, C, P, G. That is just a way in which you can represent DNA by saying that there's a phosphate group in between the A and T. But typically, you're more likely to see it like this or like this, where you don't include the phosphate group, because I mean it's implied. CPG sites, if you ever hear that terminology, that just means you got a C followed by a G, followed by C, followed by a G, followed by a C, followed by a G. CPG. That's a CPG site, cytosine phosphate guanine site. Those are actually a hotspot for mutation. Part of the reason is that cytosines often get methylated by DNA methyltransferases. And when they're methylated, if they get deaminated, they may be deaminated into thymine. And as you'll come to appreciate when we go through the repair mechanisms for DNA, one of the repair mechanisms is recognized as damage base. However, a deaminated cytosine is not a damage base, and so you'll lose that repair mechanism, and so it'll be easier to mutate a sequence that has cytosine in it. That probably did not make sense to you, and I didn't mean it to make sense. You don't need to know that, but if it ever see it on a passage, at least I introduced it to you. You'll note another thing here is that DNA always hybridizes 5' to 3', 3' to 5'. It'll only, if you're going 5' to 3', it'll only link up with a DNA strand going 3' to 5'. And they have to be complementary to one another. We can now move on to some of the, more research-based things about DNA, which is UV spectroscopy. As you know, nucleobases that are found within DNA are aromatic heterocycles containing nitrogen. Because they are aromatic, you know that they are conjugated. Because they are conjugated, and conjugated means that you have double bonds in tandem with one another. So you get a double bond, space, double bond, space, double bond within the carbons or the nitrogens. Or you have sp2 hybridization in tandem. Anyway, you get that. That means you're conjugated. And if you're conjugated, then you absorb UV light. You absorb UV light because you're able to excite electrons from the highest occupied molecular orbital to the lowest unoccupied molecular orbital, the luma, from HOMO to LUMA. And it turns out that it'll absorb light in that wavelength range. And it will absorb UV light in order to make that jump, because it turned out that UV light is just the right frequency and hence just the right energy in order to make that jump. It corresponds exactly to the energy difference between the HOMO and the LUMO. Why don't we look at this graph here? You'll find that on the x-axis we have temperature, and on the y-axis we have the absorbance of UV light at 260 nanometers. It turns out that double-stranded DNA will absorb light differently than single-stranded DNA. The rationale of that double-stranded DNA has the space stacking effect that plays a role in then the absorption of 260 of the light that is being shown onto it. We don't have to go there. We don't have to understand why that is. Let's just appreciate the fact that that is the case. That's called the hyperchromatic effect. So as DNA denatures, as DNA goes from double-stranded to single-stranded, it absorbs more UV light, and it's depicted in this figure. The TM, the melting temperature, the temperature at which 50% of the DNA is denatured. So the temperature increases, we start to denature more and more and more. As we know, temperature denatures protein, then it also denatures DNA, and it becomes single-stranded. Another thing that denatures nucleotides is going to be low, is going to be high pH, so basic pH, and you might want to consider why that is. It's actually not rocket science, and I know you can't figure it out, so I'm not going to tell you. But things that denature nucleotides are actually high pH and temperature, and chiotropic agents. The hypochromatic effect is saying the opposite, but it's the same. It's playing on the same effect. So they're both kind of opposite, polar opposites, but same thing. If you know one, you know the other. Again, the melting temperature, the temperature at which 50% of the DNA is denatured. If you're confused about why DNA absorbs UV light, you should watch the optics lecture. There I discuss how it just so happens that light of the frequency in the UV range, or the wavelength in the UV range, is resonant with the electrons that are found within the highest occupied molecular orbital of the nucleobases, of the carbon and the nitrogen of the nucleobases, and it can make the electrons jump from the HOMO to the LUMO because they are of the right energy and frequency. But again, that won't make sense to you if you haven't watched the optics lecture. Note the trend. There is an inverse relationship between hydrogen bonding and UV light absorption. If you increase hydrogen bonding, you decrease UV light absorption. Here's the nice little exercise for you. I want you to match the colors with the DNA strands. So go ahead and do it, and then I'll show you the answer. Pink. The answer is A is the pink one, B is the green one, C is the blue one. I imagine you're wondering why. So why don't we deal with the blue one first? The blue one is this guy. So clearly, if you have less GC bonds and you're smaller than the rest of the strands, then you should unhybridize, go from double-stranded to single-stranded before any of the other ones at a lower temperature than any of the other ones, because it's going to take less energy to pull you apart. Because the AT bonds aren't as strong as the GC bonds, and if you have less bonds in general, clearly that's going to be the case. Alright, how about the pink one? The pink one, if you're comparing A and B, you'll know that you have to put in more energy to break B than to break A. So clearly the pink one is going to correspond to A, because you're going to have to put less energy than you would have to do to break up B, because B has a lot of GC bonds, and we know GC bonds have one more hydrogen bond than any of the other bonds. So this is, the last one is B. There you go. Nucleic acid function. We did structure, now we're going to do function. The function of DNA is both to store genetic information and either allow or prevent access to that information via the different modifications of DNA that can occur that we've already gone through. The methylation or the acetylation. The methylation being gene silencing, and the acetylation being promoting gene expression. It is used instead of RNA because DNA is more stable than RNA. We've already gone through that as well. I told you that the 2' OH on the RNA that is not present on DNA can do a nucleophilic attack on the file-shaped backbone and then cause for the formation of discontinuous RNA or DNA. So that's why we use DNA instead of RNA. That's what makes it more stable. There it goes. What makes DNA more stable than RNA? So what would happen here is this OH here can do a nucleophilic attack onto this phosphate and call this guy to leave. It'll probably pick up a proton somewhere, so let's put a little H here. And then you don't have this, you would then break up the strand. And if you break up the strand, then you're not really maintaining the integrity of the code, because of course the polymerase, as you'll come to appreciate, has to read this read these nucleobases in order to extract the information from DNA into RNA and ultimately into protein. And if you disrupt that sequence, then you've disrupted the code, you've disrupted the information. The entirety of DNA is contained in a structure. For example, the nucleus is called the genome. If it's in the nucleus, it's called the nuclear genome. If it's in the mitochondria, it's called the mitochondrial genome. So when you talk about a genome, you're referring to all the DNA found within some structure. The area in which a sequence or sequence element or just element of interest resides is termed its locus. You'll see this terminology in the context of transcription. A sequence element, or just an element, all right, a nuclear element, is just a sequence of nucleotides of interest. And it may have some function, it may not, it may just be what you're looking at. But that's just termed this locus. A locus is simply just the area in which you're looking at. It can be defined as, X amount of genes. It can be defined as, 10,000 base pairs or 30 base pairs. When a portion of DNA is expressed, it is called the gene. There are some non-coding areas of DNA, and they're just called non-coding. But when they are coded for, when they are expressed in some way, shape, or form, or notably have some function, they're called a gene. An allele is a variation of a gene, and I'll give you a taste of that. So again, let's say this area is our area of interest. Then I can call this entire area the locus that I'm looking at. All right, and within this locus we find gene X and gene Y. Gene X in green, gene Y in yellow. I labeled this allele A. So let's say this is the gene X and it's allele A. I told you alleles are just variations of a gene. So if, let's say, in a different person or on another chromosome of this person, you find that you have a CG where you used to have an AT, then we would call that allele B. Okay, gene X, allele B. It's just a small variation of the gene. It maintains its functionality, but instead of having an AT, it has a CG. So that's what makes it allele B. It doesn't have to be a CG, it could be anything, it's just some variation. So that when you transcribe this and then you translate it, it comes out as a slightly different protein. There you go. A DNA sequence which repeats itself is called a satellite sequence or a tandem repeat sequence. So let me show you that. I've highlighted the repeat sequence in different colors so that you can see how it repeats itself. You'll see that it's A to ATG, CGG, ATG, CGG, and it keeps going like that. That's called a tandem repeat sequence. So tandem just means one after another. Repeat means clearly repeat, and it's a sequence. We could also call a satellite element. You may hear that terminology. It's just saying the same thing. You'll see this in the context of, well, there's many different types of satellite sequences in our DNA. You got long tandem repeats, short tandem repeats. So long tandem repeat just means it's a long stretch of repeating DNA. Short tandem just means it's short. And you'll see this in the context of the centromere. The centromere is a satellite sequence. It's a repeating sequence that you find. And it's used as kind of a satellite point and a binding point for a protein called the kinetochore, a complex of protein called the kinetochores that's ultimately used in meiosis and mitosis. You'll also see a satellite repeat sequence or a tandem repeat sequence at the telomere. Today we'll go through that, the telomere, and how it's used in protecting the ends of DNA, which we have a lot of trouble replicating. Anyway. A sequence that can be moved from one locus to another is termed a transposable element. So it's just like moving this from here, from its original position, gene A, over here. It can be moved. And it's not like it's moved by magic. There are enzymes that make cuts right here, and make cuts right here, and cut the phosphate backbone, and then remove it, and move it over to another area, another locus, okay, within the genome. And there it goes. So it moved from around here and down here. And the reason why everything else moves is clearly you cut something out, so everything else has to go fill it in. DNA binding proteins recognize DNA with sequence and structural specificity. So what we're going to go through next is going to be, DNA replication. And there are going to be some DNA binding proteins that bind to the DNA, and you're going to wonder how does it know to bind at that location. It's not just about the sequence. Yes, they use the sequence and they use these base pairs and the moieties found on these bases to recognize the right position to bind to, but it's also structural. They'll either bind to the minor or the major groove, okay, and they'll bind in such a way that it is important for these nucleotides and these base pairs, these nuclear bases, to be oriented in the right direction and in the right register. All I'm trying to impress upon you that is sequence-specific context. So let's say you were to tighten this twist. You tighten the twist, then this protein may not be able to bind anymore because maybe this base would now be located right here, and this base would be located right here, and then it wouldn't be able to bind. It's sequence and structural-specific context that it makes. This is actually a zinc finger domain. Kind of looks like a finger, and so that's why they call it that. You'll notice that the amino acid backbone here, as well as any side chains that are not being shown, is probably interacting with these bases and having these, positive electronic reactions with each other and not electronic repulsion. So after that, we can move on to the cell cycle, G1, S, and G2. I don't really have much to say about G1 and G2. It's mostly going to be about S phase, which is where DNA replication occurs. So here's the cell cycle. Why don't I just go through it broad stroke? Let's start at the growth phase. The growth phase, the G1 phase, the first growth phase, is just a phase in which our cells grow. Okay, they receive signals to grow, and they start to transcribe and translate protein that allows them to grow. They keep growing. They keep using their resources and their environment to ultimately grow larger in size and transcribe and translate the right proteins required for DNA replication and eventually mitosis. Okay, there's a checkpoint associated with this. The cell has to be of a certain size and not have DNA damage in order to get past this checkpoint and go into the DNA replication phase. They also have the option of not dividing ever again, and that's called senescence. We'll mention that in the context of telomeres. Anyway, if we can go into the DNA replication phase and receive the right signals from the environment, extracellular matrix, what have you, it'll replicate its DNA within S phase, and then it'll go to the G2 phase. That's another short growth phase. In that short growth phase, there's a check for cell size and DNA replication, and make sure it's not stalled, make sure the DNA is completely replicated and not damaged in any way. That's the G2 checkpoint. If you can get past that, you can go through mitosis, which remember is prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. There's also a spindle checkpoint at metaphase, and it's essentially a tension checkpoint. The microtubules that are coming out of the centrioles and binding to the kinetochore protein that is bound to the centromere, if those don't feel a tension check, if they pull on the kinetochore and they don't feel a tension feedback, then you won't get past the checkpoint. If you do, then you can go through anaphase, pull to each side, and eventually go through telophase and cytokinesis. And then you're back at G1. So we're going to focus on the S phase today. But before we get on to the S phase, I want to talk about the regulation of the cell cycle very quickly and briefly. I think I'll talk about it more in the mitosis lecture. So the cell cycle is controlled by cyclin-dependent kinases and ubiquitin ligases. I think it's relatively clear, or at least it's relatively just by the name, cyclin-dependent kinases is what they do. Kinases add phosphate groups or remove phosphate groups, particularly add phosphate groups to groups. It's a matter of perspective, but they add kinases to compounds, and they're regulated by a protein called cyclin. And these cyclins cycle in a very rhythmic manner to the cell cycle. So they cycle up and down in concentration, and when you have high levels of cyclin, then you can activate the cyclin-dependent kinase. There are many different types of cyclin: cyclin D, cyclin E, A, B, and all of those control different cyclin-dependent kinases to ultimately move us through the cell cycle in a rhythmic, coordinated manner. The other player involved is getting rid of the activated cyclin-dependent kinases when you need to, and that's what ubiquitin ligases are in the business of doing. What they'll do is they'll target the cyclin and the cyclin-dependent kinase complexes for degradation, and they'll do that through the proteasome. So I'll show you that. But you'll notice here in this graph that as this APC activity goes up, so anaphase promoting complex cyclosome, that's APC, as their concentration goes up, you'll notice that you'll see a quick drop in the cyclin-dependent kinase that it targets. So this yellow one targets this CDK1 cyclin B complex, and it will destroy that. It'll target that for the proteasome. So why don't I show you that? So I'm going to go through this very quickly. The E1, E2 ubiquitin ligase system targets proteins for degradation. When a protein is polyubiquitinated, poly now, not just one, but polyubiquitinated, that is going to target it for degradation via this proteasome. I don't care that you know all this jazz here. You should just know that if you have a polyubiquitinated protein, it's going to be targeted to the proteasome, which functions to hydrolyze polypeptides into oligopeptides. Okay, short peptide sequence. Oligo means short. And they do that just by having these amino acid residues within the barrel region of the proteasome stick out and hydrolyze and do nucleophilic attacks on the peptide bond of the polypeptide. All right, so the APCCs that control then the concentration of specific cyclin-dependent kinase cyclin complexes are the ones that will help to regulate then the cell cycle by getting rid of, destroying, targeting for degradation the cyclin-dependent kinases that were activated due to, increasing and decreasing levels of cyclins. And that's it. Just to be clear, you do not need to know what the proteasome does for the MCAT. It's not listed anywhere, but it doesn't hurt to know it either. Okay, so we can go into S phase of the cell cycle now. DNA replication. DNA replication occurs during the S phase of the cell cycle. Alright, so what we'll do here is we'll summarize DNA replication, then we'll go through it in all its raw detail, and then we'll summarize again. DNA replication is a process by which our cells makes a copy or replicates our genome. DNA replication occurs during S phase in the nucleus. DNA replication utilizes the sequence of DNA binding proteins. The origin recognition complex, or the ORC, recognizes the DNA sequence and marks the beginning of a replication fork. CDC6 and CDT1 loads the DNA helicase. CDK2 and DDK1 activate CDC6, CDT1, and MCM27. Once this has occurred, you can no longer open up any more origins. DNA polymerase alpha sets down the RNA primer. DNA polymerase epsilon and delta catalyze the polymerization or the lengthening of a nucleic acid primer via adding complementary nucleotides as per the DNA strand of the template. DNA helicase, or MCM27, catalyzes the ATP-dependent unwinding of DNA double helix so that the polymerase may read the template DNA and extend the primer. pCNA sliding clamp tethered the DNA polymerase to the primer template junction, increasing its processivity or nucleotides added per binding event. Replication factor C, or RFC, coordinates the placing of the pCNA sliding clamp and the DNA polymerase epsilon and delta onto the primer template junction. Single-stranded binding proteins, or RPA, bind to and prevent single-stranded DNA from reannealing during replication. Flap endonuclease, or FEN1, cleaves the flap DNA from the lagging strand. That flap DNA results from Pol delta plowing through the Okazaki fragments on the lagging strand. FEN1 will cleave that strand out, and then ligase will seal the nick, which is a free 3' OH and a free 5' phosphate. DNA topoisomerases work to prevent supercoils. Telomerase replicates the ends of DNA, and the shell-turn complex binds up and protects the ends of DNA. DNA replication is an energy-intensive process. Each nucleotide added uses up an NTP. DNA helicase uses up ATP. pCNA sliding clamp uses ATP. Every round of DNA replication, you double the amount of DNA you have. In n rounds of replication, you'll have 2 to the n of DNA, 2 to the n times of the DNA you originally started with. An error in DNA replication is called a mismatch. If that mismatch is not fixed by DNA polymerase or any of the other repair pathways, guess what? After 2 more rounds of replication, you'll have a mutation. DNA replication occurs within the nucleus. It also occurs within the mitochondria. Again, they have their own DNA and their own transcription and replication machinery, and it actually turns out that it's very much prokaryotic in nature. The way in which their replication occurs is prokaryotic in nature, and the way in which their ribosomes work is prokaryotic in nature. Again, they have circular DNA as opposed to our linear DNA. Everything that we go through today, you can assume, is talking about eukaryotic replication, unless I say otherwise. So any picture you see will always be eukaryotic, unless I tell you it's otherwise. There's actually one picture I put in here that's actually prokaryotic, but other than that, it's going to be eukaryotic. Anyway, here is just what the polymerase does. The polymerase does, it functions to polymerize nucleotides. It functions to connect the nucleotides together to form a polymer, this long biopolymer. And what happens is that, simply that, it'll extend a primer using its 3' OH, do a nucleophilic attack on, let's say, TTP, so thymidine triphosphate. It'll do that attack, and then it'll incorporate that thymine. Okay? And it actually does it randomly. It doesn't just pick out the right one. Actually, all these sample into the active site of Pol epsilon, and the ones that fit will get incorporated. And actually, this isn't really the best picture, because in the reality of it is that DNA replication does not just start at the end of a DNA molecule. It actually starts somewhere in the middle, and then it'll go outward using a replication fork. So that's what we're going to go through next. But I just wanted you to appreciate the fact that what we're doing is just extending this, a primer. It doesn't have to be an RNA primer, but it turns out that Pol alpha primase will actually only put down an RNA primer. So the first step in replication is that the origin has to be recognized by the origin recognition complex and the related factors. Okay, origin is where DNA replication begins. So the ORC complex will bind to the origins, and there's CDT1 and CDC6. You already know what they do. They function to place onto the primer template junction the MCM27 or the DNA helicase. So that's what they do. That's what CDT1 and CDC6 do. They'll add those on. Now, I picked these origins randomly. These are all origins. These are all places where DNA replication can begin. And one thing that you should come to appreciate very shortly is that you can only activate a few at a time. Not because it's impossible to activate them all the time. It's just the random kinetics of it. So I'll show you that in a second. So rising levels of cyclin E activate CDK2 and DDK, activating the protein and causing for phosphorylation. All right, again, I just told you the control of the cell cycles via these CDKs. Let's not really go with DDK. Ignore DDK. CDK, cyclin-dependent kinase. They'll phosphorylate these proteins, and that'll cause for the activation of these proteins. That'll dissipate the origin recognition complex and cause for the activation and release of CDC6 and CDT1 and the recruitment of proteins involved in DNA replication. So watch this, quick. Just that phosphorylation event, that is causing then all the subsequent things to occur. Okay. Well, we've brought into the game replication factor C that functions to load the PCNA sliding clamp, and we brought into the game PCNA sliding clamp. Okay, and we activated this helicase. And now, after phosphorylation event, after we've removed CDC6 and CDT1, they are so-called inactivated. They were activated in the sense that they brought up all these proteins, but they are inactivated in the sense that they cannot no longer bind to a new ORC complex. CDC6 and CDT1 after phosphorylation via cyclin-dependent kinase 2 can no longer recognize the new origin. So none of these origins are going to initiate replication fork. They're going to get replicated, but they're going to get replicated by these origins. So they're not going to initiate their own origin anymore. You can't. And then you have DNA replication. This is the messy slide. Okay, don't try to follow this slide. You can if you want, but it's actually quite confusing. And I don't expect for you to be able to read this too well unless you sit there and study it. But you'll notice you have a single strand of binding protein, the RPA, binding to the single strands. You have the PCNA sliding clamp, increasing the processivity of the DNA polymerases. You have Pol alpha primase setting down these RNA primers, and we're extending. You'll notice that Pol epsilon always goes towards the fork. Pol delta always goes the way. All right. I'm going to go into a nicer slide soon. I don't like this slide. So here, just impressing upon you the same thing that I just told you. I'm going to superimpose the phases of DNA of the cell cycle onto then what occurs during origin recognition. So here are the origins found in red, and the blue just regular DNA. So let's say the ORC complex binds to this origin, and it doesn't bind to any of this one just by chance. Then once you go into S phase and start to replicate this DNA, you're not going to get the initiation of these origins to undergo DNA replication. You're just not going to get that. So in G2, then that's what it looks like. You didn't initiate these origins. All you did is replicate from this origin. So note the trend. Once CDK2 and DDK phosphorylate CDT1 and CDC6, you may no longer begin a new replication fork. One thing that I want to mention here, just because we're talking about origins, that in prokaryotes, they only open up one origin at any time. They'll never open up more than one origin. They'll use one origin and form one replication fork, and that'll be it. Here's the passage exercise for you to do. I'm trying to take a very research-based approach in these lectures, and so I decided to incorporate a passage. I'm not going to go through it. I'll just tell you the answer. That I think it would make the lecture too long if I just read it to you and all that stupid stuff. So do it, answer it, and I'll just tell you the answers here. Alright, so the answer for 1 is B, the answer for 2 is B, and the answer for 3 is B. And since that passage went into chromatin immunoprecipitation, I think I should explain it to you. And the reason why I'm explaining this to you so that when you read about it in passages, you won't get confused. Alright, so chromatin immunoprecipitation. Chromatin immunoprecipitation is typically used to determine the sequence that DNA binding proteins bind to DNA. So they want to know if a protein binds to DNA, at what sequence does it bind to, and does it bind at all? So how do they do that? The first thing you need to do is cross-link your DNA binding protein to DNA. So what you do is you just incubate cells with formaldehyde. Formaldehyde will get into the nucleus and do a cross-linking reaction to cross-link the protein to the DNA. And actually, if you know what formaldehyde looks like, it's just the most simplest aldehyde you can ever get. Okay, it's just a carbon double bond oxygen with 2 hydrogens coming off it. It makes for then a great target for a nucleophilic attack. So that's how it cross-links. You just get nucleophilic attacks onto the formaldehyde group. Typically, it'll be imine formation, and then you'll get the imine to attack then another moiety and do a nucleophilic attack there. Anyway, you cross-link the protein to DNA. You shear the strands via sonification. You don't have to understand what that means. You just shear the strands. And then you add a bead-attached antibody to immunoprecipitate the target protein. And the reason why we have to, use an antibody to get our protein DNA complex is that when you cross-link the proteins, you didn't just cross-link your protein to DNA. You cross-link every single protein that was bound to DNA at that time. And so you need to get your antibody, you need to get your protein DNA complex out of there. So use a bead-attached antibody. So the bead, what's the bead for? The bead is just used for then when you want to separate the antibody DNA protein complex from solution. The bead is what's going to be recognized by an affinity chromatography column. And if you haven't studied that yet, maybe it doesn't make sense to you, but the affinity chromatography column will bind to the bead, and you'll separate your protein complex from the solution. And then you unlink the proteins, okay? You break up the formaldehyde link, and then you can sequence the DNA. And when you sequence the DNA, you'll find out what DNA sequence your protein binds to. And then you can map it back to the genome to find out where in the genome that location is, as well as what gene it is. That's what chromatin immunoprecipitation is used for. Alright, here's the replication fork in an easier format to digest. You've got leading strand, you've got lagging strand. Pol epsilon will go towards the fork every single time. It'll go towards the fork, alright? And Pol epsilon is always found on the leading strand. Pol delta is always found on the lagging strand. I remember that because I know delta means change, and I know there has to be a lot of changing going on with Pol delta in terms of Okazaki fragment formation and having to go back towards the fork, and we haven't gotten there yet, but we'll get there now. You got Pol epsilon, you got the PCNA sliding clamp that increases the processivity of Pol epsilon, replication factor C that helps to load the PCNA sliding clamp onto the primer template junction. You got Pol alpha primase that lays down the RNA primer. You got Pol delta that replicates the lagging strand. You got single-stranded binding protein, then you got Okazaki fragments, which are in red and blue here. The blue, the primer, red is the RNA that's been added, the DNA rather, that has been added via Pol delta. You also got MCM27 still doing its job of breaking up the hydrogen bonds that hold together this DNA, and that's using up ATP. So again, what made this structure, what helped to make this structure, the origin recognition complex, recognizing origin, loading and activating MCM27 to then start going this way. And it turned out that on the other side of this, there would also be replication occurring, but I wanted to zoom into one side of the replication fork because it makes it easier to look at. There's nothing really here that is probably confusing to you except for the fact that these Okazaki fragments and this lagging strand synthesis and why it's working this way. The reason why it's working this way is because Pol polymerase can only extend 5' to 3'. That's to read 3' to 5' and extend 5' to 3'. So look at this template strand, this black strand. So the template strand is the strand that the polymerase reads. So the template strand for the lagging strand is running 3' to 5' in this direction. So it's reading it this way. But it has to extend it 5' to 3'. Okay, so it has to go like this. And you'll notice that it has to wait for this to open. So I'll show you that. Okazaki fragments result from the DNA polymerase extending only in a 5' to 3' direction. So you'll notice I labeled the template strand in terms of what its orientation is. So this one's 5' to 3', this one's 5' to 3' this way. Alright, Pol epsilon will just go straight forward. It'll just keep going, keep going, keep going, because it's extending from 5' to 3'. And there's no hindrance to him doing that. Because as he goes, the replication fork also goes, and it opens more and more and more, and it can just keep extending. However, Pol delta, what's going to happen with Pol delta? What is going to happen? Well, as you know, Pol delta can only extend 5' to 3', just like Pol epsilon. And what will happen when we open up this replication fork is that Pol epsilon can keep going forward. However, Pol delta has to actually, now let's go back to Pol delta. Pol delta actually has to go like this. it actually has to move and remove itself from this area and come all the way back here to be able to extend again. Because this area down here is already extended. He already did that. So he needs to go back this way, okay, in order to extend again. All right? And pole epsilon can just keep going forward the way it's been going. So that's why they're the leading strand and lagging strand. The fact that the polymerases only extend 5' to 3'. And while one of the polymerases, the leading strand, can go 5' to 3' and keep going towards the fork, one has to go away from the fork in order to replicate that other strand of DNA. And if you have to go away from the fork, it's going to be discontinuous because you have to wait for the fork to open to restart your replication. Okay, so that's where Okazaki fragments come in. Okazaki fragments are just named after the researcher who researched it. And Okazaki fragments are just discontinuous strands of DNA and RNA. And those have to be removed at some point. So we'll go through that at some point. What happens is, to remove them, to remove the Okazaki fragments, Paul delta will actually plow through an Okazaki fragment. Alright, and it creates this flap here. And this flap is called a DNA RNA flap. And there's a protein called FEN1, flap endonuclease, that will cleave the DNA RNA flap. It'll actually just cleave the phosphate bond, the phosphate backbone of the DNA. And now you have a nick. And the nick looks like this. You have this phosphate group and this free OH group. This is a 5' phosphate, 3' OH. DNA ligase will be able to fix this. So DNA ligase will just cause then a nucleophilic attack on the phosphate. And actually does that by activating the phosphate. Activating it as a leaving group. It'll actually add like an AMP group around here. And that'll make it a better leaving group. And so then it can attack and cause then the AMP to leave. And that's how it sealed the nick. Now we have continuous DNA. And I should have put it on this side. Wasn't paying attention. Anyway, that's sealed nick. Now we have continuous DNA. We got rid of the Okazaki fragments. And we're good. Again, the way in which we got rid of the Okazaki fragments is Paul delta will just plow through the Okazaki fragments. It probably has some helicase domain within it that allows it to do that. It plows through. Then FEN1, flap endonuclease 1, makes a cut. Okay, and then ligase will come seal the nick, and then we have continuous DNA. So here's the next exercise for you. I want you to indicate the polarity on the template DNA and indicate which is the leading and lagging strands. Here are the answers. This is 5', this is 3', this is 3', this is 5'. How do I know that? Well, I know this is going 5' to 3', because that's just the arrow notation. That's what that means. 5' to 3'. So then clearly then this has to be going 5' to 3', because I know the rules for annealing. I know that if I'm going to anneal DNA together, one has to be going 5' to 3', the other one has to be the opposite orientation. Which one's leading, which one's lagging? I told you, going toward the fork will always be leading, going away from the fork will always be lagging. This is leading strand, this is lagging strand. This is lagging strand, this is leading strand. Yes, they are the same strand, but we're looking at the opposite forks, okay? So we make an imaginary cut here and say, okay, this one's leading, this one's lagging. Another numerical exercise, if you start with 2 pairs of DNA, how many would you have in 20 replication cycles? And I gave you a hint. I even showed you a little replication then to see what happens. The answer is 2 to the 20 times 2 times 2. I told you that it's 2 to the n, right? So if you have 20 replication cycles, it's 2 to the n, right? Because if you start out with so imagine you started out with just 1 here, so that, and then every time it would double, so you have 2 times 2 times 2 times 2, etc. So you get to 20. However, you start out with 2, so you have to multiply by 2 you multiply your answer by 2. I hope that makes sense. If it doesn't, you just need to sit down and look at this. I've said it so many different times, so many different places. Let's just summarize the players here. Again, polymerase will only read 3' to 5', extend 5' to 3'. So this is the reading of this template. Notice it's going 3' to 5', and we're extending from 5' to 3'. All right? Leading strand going toward the fork, lagging strand going away. The factors involved, you have the primer that's laid down by Pol alpha primase. Okay? You have the pCNA sliding clamp that's increasing the processivity of Pol epsilon, the amount of nucleotides added per binding event. You have replication factor C that functions to coordinate and place the pCNA sliding clamp onto the primer template junction. You have MCM27, which is the DNA ATP-dependent helicase that will catalyze then the deannealing of the DNA strands from one another in order for then the Pol epsilon and Pol delta to read the template and extend the new replicated DNA. Pol delta go away from the fork, pol epsilon goes toward the fork. Pol epsilon leading strand, pol delta lagging strand. The Okazaki fragments just result from the lagging strand and pol delta only being able to extend 5' to 3' and not being able to reach the DNA that it needs to reach because it has to wait for MCM27 to open up the DNA to do that. It has to keep going backwards and backwards and backwards. The way in which we take care of the Okazaki fragments, because we know we need to get rid of the Okazaki fragments because we know there's no RNA in our normal DNA, pol delta will actually plow through the Okazaki fragments. Flap endonuclease will cleave the flapped up DNA, the DNA RNA, and then ligase will seal the nick. And then we'll have continuous DNA on both strands. However, there are some other issues that arise and some other questions that may arise that I want to answer for you. So we're not done yet. But before we get to those questions, I'm going to show you a review video. Now, I don't know who owns the copyright to this video. It's found all over the internet, and there's not really a clear indication as to who owns the video, so I can't request the copyright. So I'll play it, and I tried to look for somebody and I couldn't, so here it is.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA. The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
The question is, why can't DNA polymerase only extend from 5' to 3'? Well, the reason is as follows, okay? The active site of the DNA polymerase has been exactly tailored to recognize and utilize a primer going in the 5' to 3' direction and a template going in the 3' to 5' direction. That is just how the active site recognizes these compounds. It doesn't recognize anything else. It cannot polymerize in any other way. And in fact, the active site is tailored exactly to position a 3' OH group, okay, proximity and orientation catalysis, to attack the alpha phosphate of the incoming NTP molecule, be it ATP, GTP, whatever. What that'll do then is it'll cause then a pyrophosphate group to leave, okay? And that's what catalyzes the formation of, or the polymerization of DNA, okay? The magnesium ions serve as cofactors. This is a 2-metal ion catalysis mechanism, and they are coordinated by aspartate residues. What the magnesium ions do is it'll stabilize oxygen, forming a partial negative charge to attack the alpha pyrophosphate, making it a better nucleophile. And it'll stabilize the pyrophosphate group leaving by stabilizing the taking on of extra charge. It has to take on extra charge when it leaves, and the magnesium ions form positive interaction with the newly formed negative charge on the pyrophosphate group that allow it for it to be able to be a better leaving group, to be able to be more stable when they leave. And so that's what catalyzes the reaction. All these have to be in the proper register for this reaction to occur. Okay? And if they're not, if you have things in the opposite rotation, they're not going to be in the right register. You're not going to get the right hydrogen bonding base pairs between the A and the T and the G and the C and the A and the U's. And you are not going to get polymerization at any appreciable rate. And that is why. There's nothing magic about it. It's just the active site is tailored to do that. That's why. So I got an exercise for you. Is DNA polymerization a spontaneous reaction in the absence of enzyme? That's the question. Yes. 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 Enzymes only increase rate, okay? They do not make a non-spontaneous reaction spontaneous. What makes a reaction spontaneous, well, what we use to tell whether a reaction is spontaneous or not is it gives free energy. If it's a negative, gives free energy, then it's a spontaneous reaction. If it's a positive, then it's not spontaneous. What an enzyme does, it just decreases the activation energy, the amount of energy you need to get over that energy hump in order to get from reactants to products. But it doesn't change the energy of the reactants or the products, okay? And so it doesn't change, it gives free energy. If enzymes don't change, it gives free energy. Then they do not change the thermodynamics of the situation. And they can't make a non-spontaneous reaction spontaneous. DNA polymerase can fix its own mistakes. So as DNA polymerase incorporates nucleotides at some random rate, it's about, I don't know, 1 in 10 to the 5th, just considering the rate at which DNA polymerase repairs. Every once in a while, it'll incorporate the wrong nucleotide. So you'll notice here we have a mismatch here. We have a C pairing with an A when you know that there's electronic repulsion right there, and that'll never work. So what happens is DNA polymerase is going to stall a little bit. It's not going to fit in the active site after it's been incorporated. If it got really lucky, and the 3' which it just happened to be, luckily, in the right position anyway, and incorporated this nucleotide, the DNA polymerase is going to sense the mismatch because it's going to bulge out a little bit. The bond distances from end to end are actually going to be longer than 11 angstroms. And they're going to say, no, that's a mismatch. And maybe it'll be like 11.8 instead of 11. I'm just guessing. And it actually has a 3' to 5' exonuclease domain. And isn't that counterintuitive? That it extends 5' to 3', but it has a 3' to 5' exonuclease domain? Yeah, it's counterintuitive. What it does is it'll remove this. It'll go this way and remove nucleotide. And it's a different domain. It's a different active site than the active site that the DNA polymerase has for its 5' to 3' extension domain. But it uses a similar mechanism. It uses a water molecule that it deprotonates using its magnesium ions to stabilize the negative charge formation. And its 2 metal ions will catalyze the leaving group leaving, which is going to be this CMP molecule. So it'll do a nucleophilic attack, and then the leaving group will leave, which is the rest of the DNA, and then we have this molecule form. So that releases the CMP. So CMP release. And now we just wasted an ATP, but that's okay. So what I'm trying to tell you here is it has the intrinsic ability to fix its mistakes. DNA polymerase has an intrinsic ability to fix its mistake. The end replication problem. The replication of the ends of the DNA presents an important little problem that if you're thinking about what we discussed in terms of lagging strand, fixing the lagging strand problem, you'll appreciate that why this is the problem. The ends of chromatids are single-stranded. So here are the telomeres. We've gone through the structure of the chromosome. This is the short arm, the P arm, and here the Q arm, the long arm. And this is the telomere pulled out. You'll notice that the telomere pulled out is single-stranded, okay? And it has this repeating long tandem repeat or the satellite sequence repeat sequence, okay? It's TTA, GGG. TTA, GGG. TTA, GGG. TTA, GGG. And it goes on that for very long. There's a lot of repeats, so, over hundreds, over thousands sometimes. And you might wonder, why is it single-stranded? Well, we'll get there. The telomeric sequence is a tandem repeat sequence at the end of DNA. It functions to protect the loss of inner DNA. Every time we replicate DNA, we lose some DNA at the end of the strand. It is thought that the shortening of telomeres is associated with cell aging and senescence, the G0 phase, or no more cell cycle. So here's the replication for, okay? Let's start with the DNA replication that we know, okay? And we're going to move out towards the ends. Well, when we get to the ends, okay, you'll notice that there's a space here. It didn't get to this far. Why do you think that is? Turns out, though, that each one of these ends that didn't get there is the lagging strand. Because, the leading strand can easily just keep going like this, and it'll get there. But the lagging strand has to keep going back like this, and I'll draw them in and assume that it was opening. The lagging strand will go like this, okay? And it'll go like this. It'll go like this. Turned out, when you get towards the end, first off, even if you were able to get a primer here, which you don't, normally a primase won't primate, the Pol alpha primates will actually not put a primer here, but even if you could get a primer there, how would you remove it? How would you remove this? How would you remove a primer that's on the end? How we remove primers is Pol delta plowing through the primer. But if there's no DNA here for Pol delta to bind behind the primer, how can you extend that? And how can you get rid of that and then use phen-1 and then ligase to seal up the nick? How can you do that? You can't. So you get an overhang. You get an overhang. You get a portion of DNA that isn't getting replicated every single time you do replication. So it turns out then that every time we do replication, we lose more and more DNA. You'll notice that now the overhang is here instead of here. You'll notice that difference. So you're losing this little bit of DNA every time. You're losing this little bit of DNA every time you do DNA replication. Again, look at how the overhang got smaller and smaller and smaller. That is associated with senescence. So it turns out every time you replicate DNA like this and then you go through mitosis, you're losing DNA unless you have something to fix this, which most cells do not have something to fix this. Like most of our somatic cells, the cells that are found within our body, they actually can't fix this problem, and they just have to deal with it. And at one point, they'll stop dividing. And one of the notions is that some cells stop dividing and they so-called age, and they don't divide anymore because they're losing their DNA, and they're losing their inner DNA. Because you can imagine if this goes, let's say, this far in, you might start losing a gene. There may be a gene A here. And if you start to lose that gene A becomes non-functional, and the cell just will die off if it continues to divide like that. Because, once you have a loss of function, you oftentimes die because you get outcompeted by your neighboring cells. So it's thought that that's associated with cell aging and senescence. We lose DNA every round of replication, but we can fix that in some cells. Not all. And it's not a complete fix either, but some cells may be able to fix it. Telomerase functions to protect inner DNA. Again, I want to stress the fact that this telomerase enzyme is not present in most cells. In fact, it's not present it's present in very little cells. But this will function to help in some cells. What telomerase will do is it'll bind to this overhang region, and it will extend it. It'll extend it using an RNA probe. So a probe is just something that hybridizes to some target DNA or RNA. So an RNA probe is within the telomerase. It'll bind to the sequence here, the telomeric sequence, and extend it. Okay, it'll extend the overhang. It'll add nucleotides like that, and then it'll translocate. It'll move over to the right and do it again, Now it's much longer, isn't it? Notice where we started with this 3', and we extended. We're all the way over here now. We have all this DNA added. Now Pol delta can get a primer there and extend and actually protect the inner portion of DNA. So what we do is we just extend the telomeric sequence, and then once we have enough sequence to get a primer down, Pol delta can fill in the primer and protect our inner DNA from damage, the DNA that actually is important to us, because this telomeric sequence, this doesn't code for anything. This is literally to protect us from our inner DNA that actually codes for something. We don't want to ruin the sequence of that inner DNA, but we don't care about this DNA. We can lose this DNA. We can chop this off for next round of replication. That won't matter to us at all. All we care about is protecting the inner DNA. That's what telomerase is. But again, it's not found in most cells. Where you will often find telomerase active is in cancer cells, because as you know, cancer cells divide very rapidly. And if they're going to divide very rapidly, they better have something to protect their DNA. Otherwise, they're going to stop dividing very rapidly very quickly. And so cancer cells will also have upregulated levels of telomerase function. DNA replication induces supercoils. When you replicate DNA, when you open up DNA via the DNA helicase here, MCM27, what will happen is you'll increase the twist on this DNA. And as you increase the twist of the DNA, there has to be a way for the DNA to relieve the stress from that twist, otherwise it will break. And the way it does that is forming supercoils. So a supercoil is just a coiled coil. As you know, DNA, the double helix, that is an example of a coil. So when you coil a coil, you call it a supercoil. There are enzymes called DNA topoisomerases that will relieve the stress, would relieve supercoils, rather. It'll relieve supercoils. It'll open up supercoils. And the reason why you need that, and the reason why it's important, is because of the fact that if you don't do that, you will have a really hard time opening up the DNA when MCM27 gets there. So you need to get rid of these so that MCM27 can open it up. And DNA topoisomers can either increase or decrease supercoiling. It turns out that you get positive supercoiling here and negative supercoiling at the ends of DNA, of the other end of DNA. And the other reason why it's supercoiled is the fact that you have this sheltering complex binding at the end of the telomeric sequence, and that protects our telomeric sequence from being recognized as damaged, because as you know, single-stranded DNA will be recognized as damaged and destroyed. The sheltering complex does is bind that up to protect that. But that also keeps it bound on both ends, and so that's what we call supercoiling. So I'm going to show you what supercoiling is in a video shortly. But that's what DNA topoisomers is. They'll decrease supercoiling.
Alright, what I want to show you here in this video is why supercoiling occurs. Or perhaps not why, but just the physical events that lead to supercoiling. So let's say we have this DNA strand. This is the headband, but it's going to represent our DNA strand, and it's not wound around itself yet. What we're going to do is we're going to make a right-handed DNA molecule by coiling this around itself. Okay. And if you look at the curvature of it, it's going up and to the right. It's hard to see from your angle, but believe me, it's a right-handed helix. And now if I keep twisting it, and this is the kind of twisting that occurs when you undergo DNA replication. If I keep twisting it, it'll become really tense and really tight. In order to relieve that tenseness and prevent the strands from breaking, it actually will begin to supercoil. Okay, so that's the supercoil happening there. Get closer, you can see that supercoiling occurs when the tightness is too tight. Okay, in order to relieve that tension. And that would only happen if the things were tethered at the end like they are with the sheltering complex. Because if you didn't have a sheltering complex, watch what happens. So you remove the tether. Look at that. No DNA. So that's why it happens, to relieve that tension, to prevent double strands from breaking.
The semi-conservative nature of replication. So back in the day that we were studying DNA and the way in which we can replicate, there were three proposed models. There was the conservative model, the semi-conservative model, and the dispersive model. The conservative model said that when you replicate DNA, the strand that will hybridize to one another will be the original strand, and then you'll have a new strand that's hybridized with the old strand, or hybridized with the new strands. It's hard to imagine that because that's not what actually happens, but that was one of the models. That the fact that when you replicate this, the new strand will bind up with the new strand, and the old strand will bind up with the old strands. The semi-conservative model said that the one new strand will bind up with one of the old strands, and one of the new strands will bind up with the old strand. That's clearly the one that is right? That's the right one. But there was also the other model, the dispersive model, where it just kind of they mix up with one another. Again, it's hard to picture and visualize, but you can imagine. That's the right model, but I want to run you through the experiment that was done to confirm this, because I want to try to get you to think like a researcher. So here's the Meselson-Stahl experiment. This is the experiment that was done to determine that the right model was the semi-conservative model. So you take E. coli colony off a plate, and you pick it with your pipette, and you put it into a beaker that's containing growth medium with the N15 isotope of nitrogen. How did that help you? Well, what it does then is over time, when all the E. coli cells start to replicate DNA, over a long period of time, all of the DNA that is contained within the E. coli will actually contain the N15 let's just say isotope, not isotype, isotope. And that isotope is actually a little bit heavier than the N14 isotope. Remember that 15 just refers to the number of protons plus neutrons. You know, nitrogen has 7 protons, so in this case it has 8 neutrons instead of 7. What you do then after some period of time is you collect the N15 E. coli's and mix them with N14 growth medium. Why do you do that? Why do you do that? The reason is because you want to determine when these E. coli undergo DNA replication, whether or not the replication will be conservative, semi-conservative, or dispersive, and how you would tell the differential weight that occurs with conservative, semi-conservative, or dispersive. So what I'm going to challenge you to do is, well, let's finish this. Then they extract the DNA from E. coli cells at sequential times corresponding to the sequential number of replication events. So what I'm saying here is that they knew how much time it took to undergo one round of DNA replication. They knew how much time it took to wait for 2 rounds of replication. They knew how long to wait for 3 rounds of replication. And they waited, and each time they waited, they'd put it in an ultracentrifuge tube, in a sodium chloride gradient, and that would separate the DNA based on size. So I've given you the first thing. So I've given you N15 DNA and what that looks like on the ultracentrifuge. Now I'm going to challenge you to draw in what it would look like, assuming that it was semi-conservative replication. Or you could even assume the other ones too if you want. That may help you a lot. And the reason why I'm challenging you to do this is because I'm trying to work your scientific method thinking. Okay, so why don't I go through this? So after one round of replication, assuming it's semi-conservative replication, so I'm cheating a little bit already because Meselson-Stahl didn't know that, what would this look like? Well, let's look at this figure. Originally, we have all N15 DNA, and then we undergo DNA replication, and we incorporate N14 nucleotides when we do the DNA replication for the new strand. So then that would mean that we have a slightly lighter DNA product at the end of it. So guess what? Guess where that band is going to be? A little bit higher. A little bit higher. Because originally we had all N15, now we have 2 molecules of it, but it's half N15, half N14. So it'd be a little bit higher. Alright, so what would it look like after 2 rounds of replication? After 2 rounds of replication, in fact, you would have 4 pairs of DNA, and of those 4 pairs, 2 of them will be completely gray, and 2 of them will be half blue, half gray. So in fact, you should have 2 bands in this thing. Alright, you have the original band, and these are corresponding to what? These are corresponding to what? These are corresponding to the original blue-grays, and these are corresponding to the completely gray. And then after 3 rounds, you'll have less and less blue and more gray. After 4 rounds, you have less and less blue, more and more gray. This increasing size band is actually corresponding to increasing concentration of precipitate. I hope that made sense. If it doesn't, you have to sit down and think about this, because this is the kind of thinking that researchers have to do. If I just tell you the answer, you don't, and I just did tell you the answer, but if you didn't even get the explanation to the answer, you have to sit down and think about this. So we've gone on a journey together through DNA replication and all of its nuances, and so now I just want to summarize one more time. DNA replication is the process by which our cells makes a copy or replicates our genome. DNA replication occurs during S phase in the nucleus. DNA replication utilizes a sequence of DNA binding proteins. The origin recognition complex recognizes DNA sequence that marks the beginning of a replication fork. The CDC6 and CDT1 proteins loads the DNA helicase. CDK2 and DDK1 activate CDC6, CDT1, and MCM27. DNA polymerase alpha sets down the RNA primer. It's not that it has to be RNA, but DNA polymerase alpha sets down RNA. DNA polymerase epsilon and delta catalyze the polymerization or the lengthening of a nucleic acid primer via adding complementary nucleotides as per the DNA strand of the template. DNA polymerase epsilon is found on the leading strand. DNA polymerase delta is found on the lagging strand. They both read 3' to 5' and extend 5' to 3'. DNA helicase, or MCM27, catalyze the ATP dependent unwinding of the DNA double helix so that the polymerase may read the template DNA and extend the primer. The pCNA sliding clamp tethers the DNA polymerase to the primer template junction, increasing its processivity or the nucleotides added per binding event. Replication factor C coordinates the placing of the pCNA sliding clamp and the DNA polymerase epsilon and delta onto the primer template junction. Single-stranded binding proteins, or RPAs, bind to and prevent single-stranded DNA from re-annealing or rehybridizing during replication. Flap endonuclease 1 or FEN1 cleaves the flap DNA from the lagging strand. The flap DNA RNA complex results from Paul Delta plowing through the Okazaki fragments, which are just DNA fragments, DNA RNA fragments. Then, after you cleave that flap, DNA ligase seals the nick. DNA topoisomerases work to prevent supercoils. Some increase positive supercoils, some decrease negative supercoils, and vice versa. But they all work in the fashion of supercoils, either increasing or decreasing supercoils. Telomerase replicates the ends of DNA, and the sheltering complex binds up and protects the ends of DNA. DNA replication is an energy-intensive process. Each nucleotide added uses up an NTP, DNA helicase uses up a ton of ATP, pCNA sliding clamp loading uses ATP. Every round of DNA replication, you double the amount of DNA you have. So, after n rounds of replication, the multiplication factor that you use is 2 to the n. So if you start out with 4, you say 4 times 2 to the n, and that's how much DNA you have. An error in DNA replication is called a mismatch. If that mismatch is not fixed by DNA polymerase or any other repair pathways after 2 more rounds of replication, it will become a mutation. That is where we're going to go with it next. We'll briefly go through mutations. I have a lot more to say about them in a different lecture. A mutation is the misreading or misincorporation of the genetic code. A mutation within a cell arises when a polymerase incorporates the wrong nucleotide into a nucleotide polymer, or the translation machinery incorporates the wrong amino acid to a growing polypeptide. We're going to talk about, obviously, the nucleus. So let's say we have this somatic cell 1, and we want to look at gene A in somatic cell 1. Let's say if we zoom into that, we'll find that this sequence there. And we'll find that in a normal cell, it looks like that, somatic cell 1. And then somatic cell 1 undergoes DNA replication, and it incorporates the mismatch in the replicated DNA. So it has an AG instead of an AT. Now, that's a mismatch, okay? We have an opportunity to fix that, right? We have the intrinsic pathway, a DNA replication, DNA polymerase, 3' to 5' exonuclease domain, that can remove the wrongly incorporated base pair, the G, and put it back into a T. How did it know which one's the mismatch? How did it know which one's template, which one's primer? Well, it actually knows in an interesting way in eukaryotes where it actually makes cuts in the DNA, and that's how it knows that the new strand, the cut up strand is the new strand. It's actually interesting, but we're not going to go there. Let's not go there. And then it'll remove the G and put in the T. Now, if it doesn't do that, and any of the other repair mechanisms doesn't fix that, it can go on to go through mitosis. And now we have a cell still with a mismatch in DNA. And if it undergoes replication again, all right, it'll have a mutation. Once this occurs, the cell doesn't know that it's wrong. The cell doesn't know that originally it was AT. All it cares about is the fact that it's not a mismatch anymore. So that's a mutation. That's going to stay there forever now, unless the cell dies, and then it won't get propagated anywhere else. Notice the difference between a mismatch and a mutation. Just because we're here, so a transition is converting a purine to another purine or a pyrimidine to another pyrimidine. So converting from A to G would be a transition. Converting from C to U would be a transition. A transverter is converting purine to pyrimidine or pyrimidine to purine. For example, converting an A to a C. So what did we do? Did we do a transition or a transversion here? We did a transversion. We converted an A to a C and a T to a G. So that's all. Notice it's random mutation. Mutation occurs 1 in every 10 billion nucleotides. Here's an exercise for you. An adenine isomerizes to form a tautomer, adenine star, which is shown here. What kind of mutation can we expect if this adenine tautomer is found on the template strand during DNA replication? Go ahead and shoot for it. Alright, so it turns out the answer is A. What's going to happen is, and this is the way I think about it, when you have this A star, what's going to get incorporated is going to be a C when you're going to do DNA replication. After DNA replication, though, after another round of DNA replication, if it keeps going like this, you'll get a GC. So you replicate it and it'll read C on the template and say, okay, I'll incorporate a G here. And that's how you get that mutation. So the answer is A. It's not B, by the way. This AT is different from B, TA, because that would mean T's on this side, A's on this side. Alright, so now we'll briefly go through DNA repair mechanisms. I'm not going to go into any molecular detail here. I'm just going to go through what recognizes what. So DNA polymerase has an intrinsic repair mechanism. It has a 3' to 5' exonuclease domain. Polymerase will use it if it's stalled by mismatch, by missing 3' OH or a bulky base. Mismatch repair, MMR, is a set of proteins that will remove mismatches and restore the original genetic information. You have base excision repair, which are sets of proteins that deal with apurinic or apyrimidinic sites, AP sites, oxidative damage or uracil bases found in DNA from the deamination of cytosine. Nucleotide excision repair is a set of proteins that will deal with bulky adducts like thymine dimers. And then you have double strand break repair. We've already gone through this. I've showed you that. It just uses the water molecule to remove the wrongly incorporated nucleotide. It releases an NMP molecule. Let's start with mismatch repair. Mismatch repair utilizes a series of proteins to excise mismatches and replace it with the correct DNA. The way it recognizes the mismatch is through this bulge. There are a set of proteins that will recognize this bulge and recognize which one's the primer strand and which one's the template strand, and cut out the DNA and just reincorporate new DNA. Now, this is actually the E. coli. This is the only time I put an E. coli figure here. The way in which E. coli recognizes or distinguishes between the old strand and the new strand is hemimethylated GAT-C sites. When it sees the hemimethylated GAT-C site, it knows that that's a newly synthesized strand. And it knows that the strand that is not methylated, okay, at the adenine residue within the GAT-C sites, it's just called GAT-C because of the sequence, it knows that that's a new strand, and it knows to cut out that strand. So what I mean by methylated, well, that's exactly what I mean. Methylated, the adenine residue is methylated right at this position. So a mismatch repair will fix that. You'll fix the mismatch. Nothing special. Basic excision repair. So basic excision repair will recognize and fix uracil bases that are found within DNA, oxidatively damaged bases, and the correct base. And it all does that. It really all comes down to forming an AP site. Because if you form an AP site, an apurinic or apyrimidinic site, a site that doesn't have the base there, notice it still has the phosphate backbone, but it just doesn't have the nuclear base. Once you get to that site, okay, then the rest of the pathway will correct the base. So basic excision repair is just a set of proteins that'll fix apurinic or apyrimidinic sites, either being at a uracil base. And base excision, it excises the base originally to form this AP site to then fix it. There's a protein called uracil glycosylase that will cleave a glycosidic bond. It's implied in the name, and that's how it gets rid of that. This pathway is a little bit more complicated. I'm not going to go there. And sometimes you randomly get hydrolysis to the glycosidic bond, a bond connecting the nuclear base to the rival sugar. And when that happens, you have an AP site, and that gets fixed by basic excision repair. Note the trend. Basic excision repair repairs DNA by excising damaged bases that are not too large, or by fixing DNA who has already been excised. That's how it works. Nucleotide excision repair is such a protein that will deal with bulky adducts like thymine dimers. Again, bulky, you want to think nucleotide excision repair. Okay? Bulky adducts like the thymine dimer. So a thymine normally has these double bonds here, but if they'll attach to one another to form this thymine dimer, that usually occurs via UV rays. This occurs via eating actually barbecue. You know, that black stuff that appears on barbecue, a lot of it has a lot of this stuff in it, and that's actually mutagenic. And it's fixed by nucleotide excision repair. I remember nucleotide excision repair and what it recognizes by NERT. That's my mnemonic, NERT. I just remember thymine dimers and other bulky adducts. So once I remember thymine dimers, I can remember bulky adducts. So NERT for nucleotide excision repair recognizing thymine dimers and other pyrimidinic dimers. Doesn't have to be thymine, but pyrimidine dimers. Also, I make the T yellow to remember that it's from the sunlight and the UV rays that come from the sunlight that often cause for the formation of pyrimidine dimers by going through a radical mechanism and breaking up of the double bonds on the thymine, then forming that pyrimidine dimer. And so if you're wondering how people get melanoma from being in the sun, well, it's exactly for this reason. People get melanoma, cancer, skin cancer, because the sunlight will cause the formation of these thymine dimers that's mutagenic. And if it goes unfixed, if it doesn't get fixed by a nucleotide excision repair, and it's the wrong gene, and you get upregulation of some gene that increases the rate at which cells divide, then you're going to get melanoma in your skin. All right, and then we have double strand break fixing. I don't have a lot to say about this. I just think I'm going to talk about it maybe in the mitosis lecture. But there's two ways to fix the double strand break. You have non-homologous end joining and homologous recombination. We've kind of gone through what those terms mean, non-homologous end joining and homologous recombination, but I'll go through it. So non-homologous end joining just means that you don't use the homologous chromosome to fix the DNA. You just kind of process the ends of the broken DNA to make sure you get rid of any damaged bases, and then you just ligate them together. Clearly, then anything between this is very likely to get deleted and not get fixed. Clearly. However, homologous end joining uses the homologous chromosome, so let's say chromosome A from dad is broken, so you'll use chromosome A from mom to fix it. And it'll use chromosome A from mom as a template to extend and fix the broken DNA. That's much more likely to result in DNA that has not lost any information. That's all you need to know. Homologous recombination is the one that's mostly used. The only time you use non-homologous end joining is when you don't have a homologous chromosome to use. Okay, so like if you're a sperm cell or something. Okay, but if you have a homologous chromosome available to you, that's what you're going to use. You're going to use homologous recombination because that's the one that is better. That's the one that will not likely lead to a deletion. And that is the DNA repair mechanism. That is the end of the lecture. I know I've gone through a lot in a short amount of time, but I know most of you prefer brevity than me making 5 lectures about all of this, because I know you can get that from somewhere else. So, peace and love, my friends.